room. I'm going to get us started. Hi, welcome everybody. Thank you for joining us. My name is Elena Lazarus, and I'm the research coordinator for the Water and Life Interface Institute, also known as WALI, and I am the moderator for today's talk. Today is the third talk in our Anhydrobiosis across organism scales and disciplines webinar series, where we've been inviting external speakers to come talk to us about their research. And today's speaker is Dr. Duncan Ryan. Duncan is a research scientist at Los Alamos National Laboratory, and prior to joining, he completed a PhD and postdoc at Colorado State University. Dr. Ryan is a nanomaterial and microscopy expert and is going to be talking to us today about quantum ghost imaging and its applications for plant biology and plant physiology. If you have questions during today's talk, please write them into the Q&A box you can find at the center of your Zoom screen. We're going to have about 10 to 15 minutes for a Q&A session once the talk concludes. And with that, I'm going to hand things over to our speaker.
Great. Thank you so much, and thank you for the invitation to talk about this work. And even in that introduction, I've actually strayed quite a bit from what my PhD work was, as nanomaterials, and now I'm doing bioimaging and quantum imaging. So it's interesting where your career ends up taking you. So yeah, what I want to talk about is kind of some new instrumentation that we've developed for the study of plants. Kind of this merging of quantum imaging techniques with actual practical biology and the needs of the biological world. Like all of us, we have to satisfy our sponsors, and one of the reasons we got this project running was kind of a DOE BER grant that wanted to study drought-tolerant plants and understand the biology, the functionality of these things better with new imaging techniques. And so there's an interesting pairing here. My background is in optical microscopy, but the application space that we're really trying to target are imaging plant samples. So since my background's in physics, I'm going to be a little bit less I'll talk a little bit less about the plant biology here, and I'll describe a little bit more about the physics of what we're doing. But the issue that we're really trying to ask is, how can you study plants and the various signatures that they have with new microscopy tools? Now, infrared spectroscopy is kind of a big umbrella term that encompasses UV-Vis, Raman, FTIR, and all these techniques really tell us about chemically what is present in a sample. And if you know what is chemically present, then you can track the chemical changes over time. That's really the point of long-term studies. Now, I'm in New Mexico, Los Alamos National Laboratories in New Mexico. We're known for our chilies, and so this is actually an interesting little study that I saw of a pepper plant. Where they did really IR spectroscopy to look at where the nitrogen was in the plants, where does it concentrate? And in particular, what's fascinating about the study was you could see nitrogen really concentrating on the upper leaves and less concentrated in the lower leaves. And of course, upper leaves are exposed more to the sun, and the more sun these plants are exposed to, the hotter the chilies tend to be. But these kind of spectroscopic signatures are really important to map spatially. So if you know where these signatures are within the plant, you can get an idea of what's happening with metabolic processes. Plants are of particular interest because there's a lot of spectroscopic kind of targets to look at. If you look at really the breakdown of the major mass components that exist in the plant, there's water, which is really the dominant feature across most of the infrared spectral region. But there are a couple other components in there that have strong spectroscopic signatures. There's the protein mass, and then the lignocellulose mass that's in there. And when you look at the absorption spectra within this kind of 1 to 2 micron range, which is kind of where I'm going to be targeting here. The various components between water and protein, they contribute different amounts to the absorption of those plant matter. And in particular, water kind of takes over pretty quickly. And so I know we're talking about desiccation and really where's water distribution within plants. And so those water features are of particular interest when you're trying to look at what spectroscopic signatures there are in the near infrared. And really do become kind of dominant when you start looking at that above 1 micron region, the water peaks there, that blue curve really does tend to have the strongest absorption coefficient among all those other mass contents that exist in plant matter. So our group is particularly interested in sorghum and camelina. And another thrust of what we've been doing is not just developing these new microscopy techniques that I'll talk about in a second, but also studying them with conventional infrared microscopy. So FTIR is really one of the more common techniques for doing this. And FTIR is great because it gives you a whole spectrum of what's going on in the plant. So you can identify specific components, features that are strong absorbers or strong reflectors in the case of this particular data that I'm presenting right here. Those peaks really do indicate specific chemical signatures. And then if you do different kind of stresses of the plant, you can look at which components of these things change depending on their stress. And in FTIR, it's pretty common to look at these principal component decompositions of which peaks are changing and which peaks are staying static. And so this is really the conventional way of looking at the absorption spectra of plants. But one key thing that is missing here is these are usually done in either powdered form or chopped up samples or some sort of non-living version of the plant. And you don't necessarily get the spatial context of where these individual components are. A leaf might have various distributions where there's a strong peak in one region, but a different peak in a different. So one method to kind of overcome that is, again, taking these conventional FTIR images. And FTIR is, a points image. So you can scan your point image across your leaf and look at the spectra as it changes across that leaf. And so you can, in theory, get different spectra at different points, but you do have to go through and scan through your plant. And this is what I call non-imaging method modality. It's where you have to recreate the measurement at every point of interest. And so if you have a sample that you really want to kind of develop the rich context of all the spatial information in there, you have to do very fine point scans, and that takes a long time to do. And then oftentimes the sheer process of putting a plant leaf into an FTIR, which requires very high light levels, especially if you're doing an absorption measurement, it requires very high intensity light to be projected onto that plant. So that plant is essentially experiencing very different environmental conditions than what it normally would, and it takes a very long time to acquire these images. So if you're trying to get a snapshot of what's going on in a plant with full spatial information, the amount of time it takes to develop all that really puts the plant into a different environment altogether. What we're really interested in doing is trying to prevent that from happening. And this is the difference of looking at a plant that might be exposed to lasers at very high intensities. It's going to react. If somebody shines a laser pointer in your eye, you're going to flinch. And if you imagine the same sort of processes are going on in a plant, you better believe that they can happen. We just don't notice them because they don't have such strong flinching mechanisms as we do. So the idea that we really want to do is to take this new category of imaging techniques that use quantum light, which has the promise of reducing the amount of light that we need to measure a sample, to take it to the very low imaging conditions, and then study the plants using quantum light. So the method that we've been pursuing most recently is something called quantum ghost imaging. There is actually a classical version of ghost imaging, and that operates somewhat differently. I'm not going to talk about that too much. But quantum ghost imaging, the basic premise is you have a correlated photon source. Sometimes this is called an entangled source. I have issue using the word entangled in a lot of these applications, but it is a form of quantum light. And you take this entangled source where you have essentially two photons that come out at the same time. One of the photons we call the signal photon just goes to a conventional imaging detector. Nothing happens to it. It just goes directly to a detector that looks like a camera. The other photon, what we call the idler photon, interacts with the sample. And then after it interacts with the sample, if it's absorbed, we never see it again. But if it transmits through, then it can be detected by a second detector called a bucket detector. And this bucket detector doesn't have to have all the spatial context that the camera does. And this actually alleviates a lot of restrictions that we have on camera-based imaging, which I'll get to in a second. But whenever that bucket detector detects a photon, it can then correlate with the imaging detector, which says, does it also detect a photon? And when photons are observed in both these detectors at the same time, you can generate an image. So what's really interesting about that is the image is coming from the imaging detector, this camera-like thing, but that photon that it detects actually never interacted with the sample. And that's really a little bit of a paradigm shift here. Typically, when we do infrared microscopy, we're looking at photons that have gone through the sample and then have to be detected later. But it's the same photon that travels through the object which has to be detected. Ghost imaging is forming an image that never actually interacted with that sample. This is a lot like playing the game Battleship, and I don't know how long this reference is going to be valid. I grew up with this game, but I don't know if kids these days ever see this game. But it's a very similar kind of process where you don't really, you have a map in front of you, and you're not really sure what your opponent or what the plant is seeing. So you call out these numbers that are just kind of random at first and ask whether or not one of those photons that hit is correlated with what the other person sees. This is the basic concept of quantum ghost imaging. So a little schematic of really what it looks like experimentally. This is a little trimmed down, but essentially we have a nonlinear crystal that generates these kind of special quantum light, these entangled photons. And then we split them. One photon goes to an imaging detector. I'm calling this NCAM. The other photon goes through our sample and then gets detected by another single photon sensitive imaging device, this SPAD, but it's a single element, so it doesn't actually develop all the spatial context. Now, the photon that goes through the sample is typically called the idler photon. It's a terminology I might slip into. And the photon that does not interact with the sample, but goes just to the camera, that's typically called the signal photon. So what is the quantum light that we're really using for this microscope? There's a lot of different forms of quantum light out there. They all actually kind of originate from the same basic process at the moment, where there are some different technologies out there that are doing different things. But a lot of them are coming from these photonic crystals that can actually take a single photon and through what's called a process of parametric down conversion, convert a single photon of one wavelength into two photons of different wavelengths. Sometimes those photons can be the same wavelength. This is called degenerate quantum light. But you can also tune it, the properties of the crystal, so you get different wavelengths out. And this is a particularly interesting property that we can take advantage of for our imaging. Remember, we're trying to do imaging in the infrared. But anyone who's looked on the websites of the companies who are trying to sell detectors in the infrared, infrared sensors are very different beasts than the sensors that we have in the visible. Visible detectors are very easy to manufacture. They have very low light or very low noise characteristics. And so we really want to try to image in the visible. But we want to be probing in the infrared, where the sample actually has chemical signatures of interest. So this is where the spontaneous parametric down conversion, this quantum light really comes into focus here, is that we can probe in the visible region where our detectors are very efficient, but we can actually be probing our sample in the infrared. So in our particular experiment, we're pumping with 400 nanometer laser, detecting kind of the green area, the 540. And then actually our idler photon interacting with the plant is actually about 1.5 microns. And that approximately correlates to this band where there's a strong absorption dip, a strong absorption peak, which translates to a transmission dip, due to water. So this is really trying to target with our wavelengths the water absorption peak in that 1.5 microns. So that's really going to be giving our contrast mechanism. Is it whether there's presence of water or not, and our specific Eidler photons are targeting that region. Now, I did say that we're trying to use a detector for the signal photons, these visible photons, that cameras are much more easy to come by in the visible than the infrared. But we actually, with ghost imaging, have a couple additional requirements between the kind of the detectors we use. Maybe a lot of us are familiar with conventional CMOS detectors or EMCCD cameras. These are really the workhorse of the kind of detector technology that most microscopy uses. But the trouble is these are frame rate devices. They capture the whole frame all at once. And because there's so many pixels, you have to read them out, essentially shut off the camera while they're reading, and you essentially accumulate photons for a certain amount of time, typically several milliseconds to hundreds of milliseconds, and then you read the data out. So you really don't have any timing information with these kind of cameras. And remember, I said ghost imaging is this correlation. We need to be able to detect two photons simultaneously, and this simultaneously is on the order of nanoseconds or sub-nanosecond layers. So a millisecond integration time does not allow us to do these kind of correlations. ICCD cameras, which is actually how ghost imaging got started, these are intensified cameras that are only really active for moments at a time. So you can trigger the activation of these cameras with nanosecond resolution. Whenever an Eidler photon comes in, you can tell it to initiate the camera. But the trouble is that these things are also, they don't actually have very good contrast. You lose a lot of kind of sensitivity when you do an ICCD version of ghost imaging. SPAD arrays maybe are a little bit more familiar technology. These are single photon avalanche diodes, which is actually what our bucket detector is. The sensor that's detecting the Eidler photon that interacts with our sample is a SPAD detector, but it's a single element. So there's no spatial context. A SPAD array essentially combines a lot of these particular sensors into a large area. They have limitations, which I won't get into technical specifications. We are actually also doing ghost imaging with SPAD arrays, but it's actually an inferior technology to something that we are going to call NCAM. And NCAM, of course, checks all these boxes. I specifically designed this chart to make it so. So, of course, it's going to be the saving grace for everything we're doing. So NCAM is a detector we've developed here at Los Alamos. I'm not part of the development team. This is actually a detector that's been developed over about 20 years by our remote sensing and group that uses this detector for national security applications. I'm in the open science part of Los Alamos, and so I actually don't do any of those applications. We're trying to take this detector for biological imaging. But NCAM, or nocturnal camera, is a single photon sensitive device. And I'll point out that this is a kind of a category of detectors called photocathode detectors. Photocathode detectors essentially are a single pixel of a photocathode material they're sensitive to single photons. Now, in our particular device, the basic operation principle is that a photon comes in, strikes this photocathode, and generates a photoelectron. That photoelectron is then amplified by a microchannel plate and takes one photoelectron, turns it into a whole cloud of billions of electrons. And then that electron cloud strikes a sensor that can record spatially where the position of that photon actually was. That's our particular process for NCAM. I will say that there are commercial versions of these detectors that are available. LinCam is one particular one being developed by a photon score. The price tag is significantly higher than what conventional microscopy detectors are, but the capabilities in our particular application really make up for this. But it gets very, very low noise characteristics. And this is really one of the advantages of using these kind of detectors for quantum imaging methods. First of all, because it's a single photon detector, we get very, very precise timing resolution. So that's sub-nanosecond, which is really what we need to do to correlate with this idler photon that's interacting with our plant. Um, they also have remarkably low dark counts. And this really, I can't understate how important this is for a lot of imaging technologies. We really think of our sensors as being very quantum efficient and having kind of the background noise of just a few hundred counts maybe. But that's per pixel when you're talking about a single or a CMOS camera or an EMCCD. These particular detectors, these NCAM detectors or photocathode detectors, have probably a few hundred counts over the entire area of the detector, which is a phenomenal difference of dark counts. So the reason why a lot of our imaging techniques, we have to increase the light levels to essentially light levels that we can see with our own eye, is because we have to overcome this noise, this intrinsic noise of the detector. With these photocathode detectors, we don't have to do that in nearly the same way. So we can tune down our light source significantly, which means we interact with our plant less. Photocathode detectors, one of the things that people look at when judging the purchase of their detector is really the quantum efficiency, the detection efficiency. And these detectors end up being kind of short-changed because they have low quantum efficiency, about 10%. But that really doesn't matter in the whole context of things when you're looking at very low dark counts. Let me give you a little bit of example. So this is actually the correlation from a ghost image. I don't have any sample in here right now, but what we generate in ghost imaging is a correlation trace. And this is essentially taking a look at every photon that came in from our bucket detector, the detector that senses after the plant, and correlating it with the arrivals of whatever came through on our imaging detector, our NCAM detector. And when there is still noise associated with that. It's much more suppressed than other detectors, but there's some noise, and so you do get kind of random events. But whenever you get a photon at exactly the same moment in time, you get a very strong peak of detection events. And this peak, if we just filter out only the photons that arrived at this moment in time, then we can generate our ghost image. So as a little bit of example here, we have that image in the center there. It says all photons. Those are all the photons that strike our center, our detector. But we have a lot of we have some dark counts. We have a synchronization laser there. You can see that pointed out. And then stray light, just because these are such sensitive detectors, they see room light pretty easily, even if you do a good job boxing it up. But if you apply this correlation, you can essentially remove all of these sources of noise and get just the correlated counts. It eliminates all these other excess sources of light. So you really do suppress a lot of noise when doing ghost imaging. So ghost imaging as a correlation technique suppresses noise in your detector. A little bit of an example of kind of why what we're doing here is an improvement over previous ghost imaging is we've been able to access much shorter correlation times than previous detectors. So these detectors tend to produce individual photons, not kind of pixelated things. So kind of the images here on the top and bottom on the left side, each spot there is essentially a detected photon. And if you look at the previous ghost imaging attempts, is you have a lot of photons, but there's a lot of kind of lack of contrast there. But with ghost imaging with our particular detectors, we can trim that down to get to contrast levels much lower than were previously possible with quantum ghost imaging. The reason why quantum ghost imaging never saw a very strong kind of pickup by biological imaging is because there's not the contrast in there beyond the simple binary targets, which is how it had previously been demonstrated. But with this kind of correlation that we're able to do, we can get down to high contrast, or we can get down to very low contrast elements of just a few percent, which is really the window you need to start applying this to real biological applications. So heralding efficiency is really the term we use for the number of photons that are collected as a percentage of the number of photons that are actually correlated with the other detector. So you actually detect a lot more photons on these detectors that will ultimately contribute to your image. And this is because of the few noise events that do occur, but also because there are uncorrelated photons that happen. But the reason I bring this up is because the heralding efficiency is just a few percent. So already I was hesitant to say these detectors are just a 10% efficient. And then on top of that 10% efficiency, you're only getting maybe 2% on top of that. So somebody said, oh, well, that's less than a percent. That's useless for biological imaging. It does create a little bit of difficulty in terms of building up an image because we do have to integrate for a long time because of these factors, but we're also trying to access very low transmission samples while not disturbing. So I want to point out that when we first got this working, it was actually in January in 2003. Um, I just wanted to point out this is my first ghost image on this particular setup. And uh, there's not much there. But I think it's important, at least for my ego, to say, look, I worked really hard to try to get the first ghost image. It didn't look like much. But then after a while, we got better at it and started actually looking at real samples that had real interest. So at first we did do some biological imaging, just fungi. There's not a whole lot going on here. This is just kind of an equivalent black and white. Where is the fungus growing on the slide and where is it not? And it, pretty well matches up with what we see in a visible image. Now, this is a microscopy technique, so microscopists like to know what is the resolution, what can you see on these instruments. I will say that in our current implementation, a lot of the data I'm going to present, we're talking about a particular imaging configuration. It can be changed to reach higher magnification. So that's actually the resolution I'm reporting. It's not the capabilities of the method, it's just the capabilities we were working with when we decided to look at a particular magnification. So we're looking at essentially resolutions of about 14 microns over a field of view of about almost 300 microns. So we're able to kind of get into that almost single cell resolution level. We're a little bit above it, but I can tell you we've already moved on from the resolution that I'm reporting here, and we're kind of subcellular at the moment. So the imaging technique is able to get to these higher magnifications and higher resolution. What's really limiting it is the fact that you still have an infrared photon, and the infrared photon can only be focused so tight. Infrared, so it's essentially the diffraction limit of the infrared photon, which determines the resolution you're going to be able to achieve here. So, like I said, we demonstrated ghost imaging last year, almost 2 years ago. Then we advanced more complicated targets. Everyone loves to see some sort of pictures. It's quirky about ghost imaging. In fact, this is quantum light. Schrodinger's cat always pops up. But what I will say is these are the kinds of images. So the ghost imaging is the one on the left, and this pair is actually an infrared camera, which shows the real thing. And this is what a lot of people who do ghost imaging have reported. They were just looking at these simple binary targets. Very hard to actually say biology can be studied if you're just looking at these black and white images, which are essentially 100% transmission or zero transmission. That's not useful for biology, which has kind of absorption transmission differences of a few percent already below a few percent. So what happens when we actually stick, say, something like sorghum sample in there? This is very obvious that we're just going to put the plant in there and see what happens. So you can see part of the microscope there. All we did is put the leaf in the field of view and then recorded a ghost image. The ghost image here, first of all, I'm showing a transmission contrast between 6% down to a background of about 1%. So this is very different than that binary target that I just showed before, which is 100% or 0%. This is 6% down to 1%. And you can see these little kind of spots, these regions of high transmission over background of lower transmission. These line up very well with the stomata of these of plant leaves. So they're very, in sorghum, stomata tend to line up in very linear patterns separated by they look like railroad tracks. And I'll say this is actually the first imaging that we did of a real biological sample, and the first real biological imaging that quantum ghost imaging has ever done. a little bit of what we get as a microscope out of this. So the raw ghost image is actually individual photons that strike in different locations. You have to convert that essentially to a transmission ghost image by taking a background. But what's interesting about these detectors is they're actually not pixelated. So you don't have individual pixels, unlike conventional microscopes or conventional cameras that do. So to make our ghost image look more like a conventional image, we actually bin them and turn them into these kind of conventional microscopy images. That's where we get the transmission of images. But I will say, so these images between the transmission ghost image and then the conventional microscopy images, there are a lot of features that line up pretty well. But the conventional microscopy images, I had to essentially turn up the light levels to the point that I could see the light, that I could place my hand in there and see the light on my hand in order to capture these. And the reason why ghost imaging works so well is that they can construct these images at the single photon level. Conventional cameras cannot because they have to have a signal that overcomes the noise level of the detector. That's really why in microscopy we have to turn up our intensity of our light so significantly because our detectors are essentially limited. Um, so one of the samples we looked at, cilantro, this is actually, we grew our cilantro. We didn't just pop into a store. I was impressed with our biologist who actually wanted to go the extra step and like, no, no, we have to do this in a controlled manner. But this is actually a ghost image of cilantro. So the difference between the conventional IR camera and the ghost image is you don't see all the features. And there's actually a reason for this. So our ghost imaging, it took about 40 minutes to acquire that ghost image. So I took a picture of the um, the cilantro leaf before, and that's actually my reference. And I also actually took an image of it afterwards and compared the difference of the cilantro before 40 minutes of being in complete darkness and afterwards. When you put the plant in darkness, after about 40 minutes, the stomata close. So that image on the bottom is actually showing the difference of intensity between the image that I took before and the image I took afterwards. And there's a reduction. Those blue spots are essentially regions where there's less transmission. This is actually the plant reacting to the environment over the course of the measurement. Because our light levels are so incredibly low, the stomata close. And we can actually see that. We're actually imaging the plant while it's doing its own natural living. Its own processes are going on. We are not interfering with that. So when you're talking about a microscopy method that might interfere, anything that has high light levels is going to be a problem because it's going to be interacting with that sample and essentially causing it to change at the time. Quantum ghost imaging is such low light. It's actually 30 times less intensity than a plant would experience if it were in just under the stars, a moonless night, just the stars. And I think this is a really powerful concept to kind of contemplate, is that if you have an imaging method which does not disturb what you are doing, does not create a dynamic or induce a biological process by the sheer act of trying to measure it, then you can actually really tune your measurements down to what is the plant doing. And, we're talking about very low light conditions, but we could also throw a lamp in there, or we could let the plant experience its full cycle of light and dark and the transitions there and actually see essentially the circadian patterns of the plant with some sort of external light source. But our measurement is not going to be inducing the changes. We can actually allow the sunlight and the darkness of night to actually create the processes that we're trying to study. Now, the real connection here, I think, between what your group is interested in talking about, really the kind of the where's the water content and where's the lack of water content in plant matter. So ghost imaging, as we've prepared it here, was really targeting the water absorption peak to make that contrast. It's essentially looking at contrast between where is water and where is not water. And that's what these pictures really are transmission focusing on that water absorption peak. So I think this is really a prime method to start looking at where are these drought tolerant processes going on in living plants. So I did just extol the virtues of trying to say, we really don't, or we're really not disturbing our plants at all. And in fact, I once had a biologist come up to me looking at my posters like, well, do you really need to go that low with light? Ironically, I saw that person make a presentation an hour later with confocal where she was saying, yeah, half of our samples burned under the confocal. So that's what happens. But do you really have to go 30 times less than starlight? The short answer is no. You could definitely be at just starlight. But at some point, ghost imaging isn't necessarily the best technique um, because we're limited by what our detectors actually have a maximum count rate. We can't exceed that. So we actually can't turn up the light any farther. It's almost the opposite problem you have on conventional microscopes, where you can't turn the light down any lower because you won't get any signal. Our problem is we can't turn the light up any higher and record images either faster or get better contrast. A little bit of example here. This is an Arabidopsis seed. These are very thin, flat seeds. And I mean, I can tell you where the outline of that seed is, right? Um, but I can't tell you the internal structure of what's going on there. I think I could if I allowed this seed to integrate over maybe a day, which is a long time to make a measurement. People are okay with that. This may be fine, but I don't really see much contrast of what's internal structure of that seed. And that's really because it's a strongly absorbing sample. And typically our process for getting around strongly absorbing samples is to crank up the power of our light source until we finally can. Another problem with ghost imaging, now I'm being honest here. I don't want to say that ghost imaging is the solution to everything, um, is that we really can't detect past 2 microns. So we can't target our absorption features beyond 2 micron because we still need that bucket detector to detect a photon. And our bucket detectors are still limited in the infrared region. Once you go beyond 2 micron, we really don't have many options. And another big problem is that this is really, at the moment, a single photon or single wavelength technique. I offered in the beginning here a comparison against FTIR, which is a spectral technique. You get the whole spectrum of absorption of your sample. Quantum ghost imaging at the moment is only operating on a single wavelength at a time. So let's talk about really what is the feature of this. And sorry, I did play a little bit too much with maybe some of these AI generators. This is in my mind what I do in my lab. I have a better beard. But what we want to do is use quantum light to get away from some of these limitations that ghost imaging is having. So ghost imaging, like I said, does need to detect the infrared photon, and at best, even better technology. So these, I'm putting the kind of the efficiency graph of single superconducting nanowire single photon detectors. These are actually the limit, the cutting edge of detectors that we could use for bucket detectors. We're actually not using them at the moment, but they're the cutting edge. And they start to cut out at around 2 micron. And after that, there's no technology. So detecting that infrared photon is still the limiting factor of this. So there is a solution that can solve essentially the spectral range problem, then also the bandwidth problem. There's a technique called quantum imaging with undetected photons. At the moment, it hasn't really been applied for microscopy. And it hasn't really been extended to broad spectral absorption measurements. But that's actually what we're working on right now. So if you could envision, so this image on the left here, it's actually material science. I'm not sure what the sample was. I grabbed it from the paper. But what they did is essentially do FTIR using an AFM probe. And they get spatial information and very good spectral information but they have to essentially sit there and move to different locations. This is that problem I kind of addressed in the very beginning with FTIRs. You have to map out these FTIR techniques. So if you want any spatial context, you have to move around a lot and require a lot of measurements, and that's time consuming. But I will say there's a way of adapting this quantum imaging with undetected photons to do these kind of measurements. And that's actually the next step and what we're working on right now. So maybe invite me back in a couple of years. I don't want to share too much on how we're going about solving that. So I'm actually a member of the Center for Integrated Nanotechnologies. This is a user facility that is housed in Los Alamos. So all of our techniques and all of our instrumentation is actually available for the public. We actually partner with uh, academic researchers, industry researchers, anyone. And the idea is that just that as long as it's an open science publication, we are interested in helping you out and working with you. We have EM facilities, we have advanced manufacturing, we have nanoscience and nanoprinting, nanolithography, and this ghost imaging, and soon, hopefully, this quantum imaging with undetected photons, these microscopy techniques that are very, specialized and not easy to come by. At the Center for Integrated Nanotechnologies, we're actually an open research institution where you can come essentially free of charge. You just write a user proposal and can come and use the expertise of our scientists and the technologies that we have available. So if you're interested, please get in touch with me about that. So I'm going to just wrap up a presentation here. Just thank the various team members. I know there's no pictures or really, I'm sure most people don't know who these individuals are, but Jim Warner is the PI who brought the money in to really develop these quantum imaging techniques. And then between Christina and point out Rebecca and Dean, Katie Seitz has done a lot of work. She's our staff biologist, I'm going to call. Really contributed to this. So it's been a huge team effort to try to develop these new imaging techniques. And I'm just the one that got lucky enough to be invited. So just want to close. If anyone is interested in chatting with me about this kind of technique or just quantum imaging and where we can go in applications for it in the life sciences, please reach out. And yeah, with that, if anyone has any questions, I'd be happy to answer anything.
Thank you so much for that great presentation. I just want to remind people you can type questions into the Q&A chat. If you want to raise your hand and ask your question yourself, I can unmute you. I see we already have one question coming in from Dr. Sue Ree, so I'm going to go ahead and unmute her.
Hey, great. Thank you so much, Duncan. This was amazing. Thanks so much for doing this. I don't know if my question is going to necessarily be directly related to quantum ghost imaging, but I was really intrigued about your last slide about quantum FTIR imaging. And I was wondering, for imaging spatially where water might be traveling in the seed while it's either drying or rehydrating. Right now, I mean, I guess we don't have to worry about a lot of light. So do you think that we could try near infrared light imaging, or what would you suggest? I mean, I guess we tried the quantum ghost imaging, and as you've very well articulated, the technology isn't quite there yet for this kind of sample, it sounds like.
Yeah, so it's interesting to see the mixture of these microscopy techniques. Quantum imaging for microscopy actually has another partner in remote sensing. So these are folks who are trying to do measurements through satellites to measure, large fields of view, or just essentially very large standoff distances. So if you're talking about trying to, say, map water content in a very large area, I think there is potential for something like these FTIR mapping, these quantum FTIR methods in a larger setting. That development, I think, is lagging behind quite a bit than, say, what the microscopists are trying to do. And I'll say I am probably one of the few microscopists who are actually trying to take quantum or trying to create quantum FTIR. Um, the field is just not populated by many people trying to do that. But the remote sensing community has been interested in trying to do these kind of standoff measurements in the infrared, where there's some advantages for looking at chemical signatures, but also because you can overcome a lot of scattering and events which degrade your signal. So there is progress, but it's these two communities that have historically not talked to one another very much. And then when you start talking about what biologically, or say, if you're trying to study a rainforest or something like that, I think those conversations aren't even being had more broadly.
Yeah,
Is
that's
that an
amazing.
answer for what you were looking for?
Yeah, that's a really great answer, actually. Also, maybe even astronomers who are
Yes.
trying to look very far out with very small amount of light for a lot of
Actually, as an
time.
interesting aside, I was speaking to an astronomer who has actually 6 of these special cameras I'm talking about, and they're looking at kind of the quantum properties of 2 different stars from 2 different galaxies
Mhm.
and how they're correlated. It's fascinating, but astronomers are using very similar techniques. Yes.
Mhm. Yeah. What do you think about like deuterium based imaging then? Just I guess what would that be called? Like
So, because
NMR? Like a
yeah, so I'm not too familiar. I have seen that deuterium is used as a contrast agent for NMR. Um, so I don't know if spectroscopically, if there's much difference between water and deuterium in terms of where the absorption peaks are. I just don't have it on top of my head what that looks like. Um, but I think I've seen a couple people using different quantum sensing methods, not these light-based ones, but kind of magnetic sensing
Mhm.
uh, for deuterium measurements.
Mhm. Yeah.
It's a category of quantum imaging that I'm not as familiar with, but I could follow up with some people who are working on that.
Thank you.
Yeah.
Great. We have another question. Um,
I have 2 questions. One is probably try to get a clarity. You mentioned that the quantum imaging cannot image anything beyond 2 microns. I'm trying to understand whether the 2 micron meaning the wavelengths or the sample thickness.
Sure. So I apologize for that. So 2 micron wavelength, and that's just ghost imaging. But this other technique, quantum imaging with undetected photons, can actually image all the way out to 5 microns of wavelength. The resolution limitations are really the diffraction limit of the infrared photon. So infrared photons tend to have a higher diffraction limit than visible photons. So that's what limits your resolution. Um, but our resolution right now is about 10 microns. We're actually a little bit lower than that right now, but it's the spectral resolution, or sorry, the photons are limited by the 2 micron, the wavelength of the photons.
What about the limitation for the sample thickness?
So the thickness is how strong of an absorber it is. So if it absorbs um, by, if it is spitballing here, if it absorbs 1,000 times, then for every single photon that goes in, you only recover one. So that's a huge contrast problem. And eventually your noise does creep in and limit you there. So 1,000 to 1 kind of transmission difference um, is is a limiting factor. But I say maybe how much is being transmitted, because obviously a thicker sample is going to transmit less. And so it depends what's in your sample. So if you have a sample which doesn't have many absorbers, you can have a very, very thick sample of it. If you have a very absorbing sample, it has to be pretty thin. So I think most of the plant leaves that we've been studying are on the order of 100 microns. That Arabidopsis seed I tried to show was probably a couple hundred microns in thickness, but much thinner can easily be done. Um, it really depends on what is absorbing and the path length of how much it absorbs over that thickness.
So second question is what other broader applications of this quantum imaging? For example, let's say uh, with other organism. One particular example is that if, since we are talking about the water desiccation stress, if I like to measure the water content decrease during desiccation, during the process of desiccation, is that something we can do, use your technology?
So it depends how so I'm thinking of one thing, kind of depending on how so I'm picturing like you have a sample and we're kind of coming in orthogonal to that. So the whole plane is essentially absorbing. But if you're looking at water molecules saying coming off of your plane, then we would actually have to turn our sample and look that way. I do think that there is interest or there's application there um, of looking at, say, the density of water at the surface. The depth of the water, you're either mounting it this way or you're mounting it this way for ghost imaging. Um, the, the, the depth is not captured because you're usually integrating throughout the entire thickness. It is an integration of absorption over the entire thickness of your sample. But if you're trying to look at water vapor levels above the, say, a leaf sample, that might be something of interest. I haven't tried that, but there might be something there. Ultimately, what it is you need to absorb a certain number of photons. So if you have such low vapor that you don't absorb very many photons, you don't really have the contrast there. But if there are enough water molecules to absorb significantly, then you would have a contrast mechanism.
Thank you. Thank you for
Yeah.
your fascinating talk.
Great. Thank you for interest.
Yes, thank you so much. It's been a great talk and really thoughtful questions and answers. So wait to see if there's an additional question. People can either raise their hand and I can unmute you, or you can put your questions in the chat. While we wait for another question to roll in, I do want to just make a couple reminder announcements. We are hosting talks once a month with the Water and Life Interface Institute in a public setting, inviting external speakers. This is our last talk for this year, but we hope to see you back in early 2025. You can follow us on social media or get on our mailing list. You can visit our website to sign up for that, to hear about our future talks, like this amazing one we had today. So, all right, I don't see anybody jumping at that chance. So I think just a round of applause and a big thank you so much for all of us for coming and sharing this really interesting technique and answering our questions. So